I'm Robin Patel from the Division of Clinical Microbiology at Mayo Clinic in Rochester, Minnesota.
I'm Nicholas Chia with the Microbiome Program as part of the Center for Individualized Medicine.
We're here to talk about our article, Pertussis Outbreak, Southeastern Minnesota 2012, which is published in the Mayo Clinic Proceedings. Bordetella pertussis and, to a lesser extent, Bordetella parapertussis cause a disease called pertussis or whooping cough. There's been a vaccine available for Bordetella pertussis since the 1940s. That vaccine was initially a whole cell pertussis vaccine. That is, the entire cells of Bordetella pertussis were inactivated either thermally or chemically. And that vaccine was very effective. However, it was associated with some side effects, including local reactions, fevers, and febrile seizures. And so it has been replaced with the acellular pertussis vaccine. At Mayo Clinic in Rochester, Minnesota, we've been diagnosing Bordetella pertussis as well as Bordetella parapertussis using PCR since 1995. As a result, we've been able to track the local epidemiology of pertussis very closely. In 2012, we had the largest outbreak of pertussis in our region in Olmsted County, Minnesota, in recent history. This is especially noteworthy because in Olmsted County, our residents are known to have relatively high vaccination rates against pertussis. The study was divided into two parts. In the first part of the study, we looked at all patients who had positive PCR results for Bordetella pertussis or Bordetella parapertussis in 2012. There were 160 patients who had positive PCR results for Bordetella pertussis and 21 who had positive PCR results for Bordetella parapertussis. We looked at the clinical characteristics as well as the vaccine history of those that tested positive for Bordetella pertussis. And what we found was that the mean age of those patients who tested positive for Bordetella pertussis versus those who tested negative was much younger. So those who tested positive had a mean age of 15.6 years, and those who tested negative had a mean age of 25.5 years. Fewer patients who had positive results for Bordetella pertussis than had negative results for Bordetella pertussis had received the whole cell pertussis vaccine. That's the older vaccine, suggesting that that older vaccine was more effective than the newer acellular pertussis vaccines. And then among those who had exclusively received the newer acellular pertussis vaccine, the time from their most recent vaccination to their positive PCR result was longer in those with positive PCR results than in those with negative PCR results. And what that suggests is that there is waning immunity to the vaccination. In the second part of the study, we did something that we haven't had the opportunity to do before. We took 265 swabs that were being submitted for testing for Bordetella pertussis and Bordetella parapertussis. And we tested them using our PCR assay, which is our routine diagnostic method, but we also subjected them to culture. We haven't had the opportunity to do a lot of culturing for this organism since we don't see this disease very often, or at least hadn't prior to this outbreak. What we found was that 12 of them tested positive by culture and 25 tested positive by PCR, showing us that the PCR is much more sensitive than the culture. We also run our PCR using what's called real-time PCR, and what we saw was the specimens that were both PCR as well as culture positive had a stronger signal, an earlier crossing point on our PCR assay than those that were just PCR positive but culture negative, again suggesting that the PCR assay is much more sensitive. Now we were able to recover isolates of Bordetella pertussis from 12 of our patients, and so we did some additional studies on those 12 isolates, which are very interesting. The treatment for Bordetella pertussis is to use a macrolide such as azithromycin to treat those patients, and rarely there has been resistance to macrolides reported. So we looked to see whether any of these 12 isolates was resistant to macrolides, and we did this using phenotypic susceptibility testing as well as genotypic susceptibility testing. And all of them were susceptible to azithromycin. So this wasn't a problem of antimicrobial resistance. We then went on to do some sophisticated molecular analysis, and I was fortunate to have the collaboration of my colleague, Nick Chia, to help me out with this.
So whole genome sequencing is a direct view into the microbial evolution of these pertussis species. And through that, we were able to uncover some interesting results that were not uncovered initially by the PCR alone, and we were able to understand the different relationships between the organisms in this outbreak.
So what we found is that we had multiple strains of Bordetella pertussis that were part of this outbreak. Now, normally when you think of an outbreak, you think about a single strain that's causing problems. And it was a bit surprising to us to see that we had multiple strains of Bordetella pertussis. This is not entirely novel. It has been reported in other outbreaks, but nevertheless, it is somewhat surprising. We also looked at what's called the pertactin type, and we were surprised to see that we had two types of pertactin within our isolates. Even more surprising to us was that when we looked at the gene for pertactin, in all cases it was altered to such an extent that it wouldn't be predicted to be expressed and produced an active pertactin protein. We had a couple of interesting other findings from this study. First, we had 21 cases of Bordetella parapertussis. This is a cause of whooping cough, but we normally think of Bordetella pertussis as being the big player. It was a little bit surprising to see that there was an increased number of cases of Bordetella parapertussis as well as Bordetella pertussis going on at the same time. Now, the vaccine, whether it's whole cell or acellular pertussis vaccine, is not active against Bordetella parapertussis. And so what you might predict is that it would be a different group of people that would be susceptible to this. And that's exactly what we found. These patients with Bordetella parapertussis were younger. Their mean age was 3.8 years, so these were little children. The mean age, recall, of the patients with Bordetella pertussis was 15.6 years. All of the patients with Bordetella parapertussis had been fully vaccinated against pertussis, again, just telling us that the vaccine doesn't work against this organism. Why this other organism was co-circulating during an outbreak of pertussis remains unclear and is the subject of future studies. Another interesting finding, and I think this is very encouraging, is what we saw amongst the very, very young. We actually had a very small number of cases amongst those who were less than 1 year of age. And we didn't see any cases in neonates in our study. And this is very encouraging. So this was more an outbreak in older children, adolescents, and young adults, which is fortunate because they get sick, but they don't end up being terribly sick. So what are the reasons why this might be the case? Well, interestingly, in 2011, the Advisory Committee on Immunization Practices of the Centers for Disease Control and Prevention changed the recommendations for vaccination of pregnant women. And what they did is they recommended initially vaccine dose during pregnancy, during the late second trimester, the third trimester, early in the postnatal period for those who hadn't been vaccinated, and then subsequently that was upgraded to any pregnant woman would get another Bordetella pertussis immunization during pregnancy. It's possible that what our data is showing us is that that strategy is working. So we saw a large outbreak of pertussis in 2012 in Olmsted County, and we did a fairly in-depth investigation of what had happened. Well, we documented genetic changes in the isolates, and this suggests that there may be vaccine escape. What I think we can also learn from this study is that we will continue to see cases of whooping cough into the future. We typically expect to see peaks of activity in the late summer, and we might expect to see epidemics that will come back every 2 to 5 years.
We hope you found this presentation from the content of Mayo Clinic Proceedings valuable. Our journal's mission is to promote the best interests of patients by advancing the knowledge and professionalism of the physician community. If you are interested in more information about us, our homepage is www.mayoclinicproceedings.org. There you will find access information for our social media content, such as additional videos on our YouTube channel or journal updates on Facebook. You can also follow us on Twitter. More information about healthcare at Mayo Clinic is available at www.mayoclinic.com. This video content is copyrighted by Mayo Foundation for Medical Education and Research.